So I would like to start by saying thank you to the organizers for letting me moderate this panel full of people that I deeply respect, and to welcome all of you panelists that have joined today, and also all the attendees which are here to listen to our discussion and possibly ask their interesting questions. I would like to start by saying that we are fully on schedule. So we have approximately 1 hour with 4 panelists. So I ask kindly all of you to keep it to more or less 10 minutes for your talks, so that we have 15-20 minutes at the ending to discuss and to address the most interesting questions coming from the chat room. The panel is an expert panel on COVID-19 diagnostics, and joining me here today are our 4 panelists, which are Dr. Alexander Lezhava, who is Associate Director at the Genome Institute of Singapore, Erik Steinfelder, who is the Biobanking Market Development Director at Thermo Fischer Scientific, Dr. Theodor Zamfirov is the CEO of Elta 90, who is one of the sponsors of today's event and one of the largest biotech dealers in Bulgaria and in the Balkan region. And last but not least, here is also Professor Radka Argirova, who is one of the most prominent and well-respected virologists in Bulgaria, and has a lot of hands-on experience during this crisis and her work at one of the biggest private hospitals in Bulgaria. So hello to all of you. Maybe Professor Argirova could also turn on her camera. And in the meantime, I suggest, good, very well, we can see you. Hello.
Yes, hello.
Thanks to everybody once again for joining us, and I suggest to give the word to Dr. Lezhava, who can share his experience in Singapore and give us more details what his involvement in the COVID situation has been since the beginning, and maybe share some valuable experience which we can all learn from. Dr. Lezhava will give you the word once you see the ball. You can start even now, because I think you don't have any slides, so you can start right away.
Yeah, thank you very much, Valentin. I think, first of all, thanks again for inviting me in this kind of interesting event, which is first time I'm participating like online, and this is I see it's quite effective, and the interaction is quite excellent according to my previous discussion when I had the opportunity. So I think I don't know if people already heard my presentation, brief presentation, but I will be brief again. I represent the Genome Institute of Singapore, and when this pandemic started, who else and not genomics people should help and should support the society when there is a need, and with our knowledge or our know-how in genomics. So beside our scientific knowledge, I am running also a translation research lab and also clinical lab, and this know-how was quite useful to come up with follow MOH recommendations, and we built a diagnostic lab here in our institute, and quite successfully. But successfully means that we had also, there was a big rush, and many people volunteered, and they finally, with quite regulated workflow establishment and various, we went through various validation and infrastructure and validation of the kit, selection of the kit, validation of the kit, also selection and validation extraction system. And then finally, we came up with a very nice infrastructure and one nice workflow. And right now, we are running on two locations. We built it. One location is running 1,000 tests, and another one is slowly evolving. And finally, we want to hit around 10,000 tests a day. So I think country-wise, Singapore is a small city and country in a small island, and with a population of about close to 6 million. And we, government took excellent measures. We managed to monitor, take measures for isolation of individuals, and I think this was quite effective. I already mentioned in previous talk that there was a very big spike once when infection hit dormitories, and that where the thousands of workers are living there, and I think that was the biggest kind of explosion, let's say. And but however, government took the measures and they isolated those areas and restricted, and then this nowadays today and these days are cases are very low, and many cases, some cases are coming from abroad, people are returning or traveling still, and those cases maybe five were imported cases today or yesterday, and in total it was 15 a day. So this is quite good number for our country, this for Singapore. And on the same time, healthcare is also on a high level from 60,000, close to 60,000 total, 57 around total population. Only 27 around there, quite low mortality we have. So it's also significant to mention. And screening-wise, I think we are going for more for population screening. I think this is a aim for near coming future, coming days, let's say. And we want to identify and isolate those positive cases which are coming up after one or another test. So I think briefly this is I wanted to mention, and later I will ask questions. Thank you very much.
Good. Thank you very much. I have my own questions to you, but I believe first we should give the floor to all the panelists, and we shall make the Q&A session afterwards. You know, I have been to Singapore once, and my impression from Singapore is, let's say, still very vivid, and I'm sure that the government has done a fine job in this situation. But I will ask you some more technical stuff later on, after we see what the other panelists have to say. So now I suggest to give the floor to Mr. Steinfelder to share his experience in the COVID crisis, and what his collaboration with scientists and collaborators around him has, let's say, produced as good practices, what challenges you can share. The floor is yours. Please proceed, Erik.
Thanks very much, Valentin. I always need to make sure that you are seeing the right screen, because I've got two. So do you see the presentation, or do you also see my notes?
We can see also your notes.
So you see that I'm already thanking you. So this should be better.
Yes, this is only the presentation.
Yeah, thanks very much. First of all, thanks, Valentin, for inviting me to this panel and be part of it. So really exciting. I think for all of us, it's getting the new normal to have virtual events like this, but it's good that we can connect, and I think that's the most important thing. I would like to stress a little bit about, from a company perspective, what we have experienced since the COVID pandemic started. I heard the word sleepless nights, and we had a rush already from the previous speaker. That's not really something that we can elaborate to. So I will just try to explain a little bit more. In the introduction, you also said that I'm the Biobank Market Development Director, which is true. So although I know what's going on, and we do work a lot in collecting positive samples for later on research, from a virology perspective, I'm not a specialist. So, but I will share and do my best. Again, I think, like many other speakers, if you are working in this environment, please be safe. I'm based in the Netherlands. Numbers are going up again. We don't know what's going to happen, but it's going to have an effect for quite a long time, I guess. So, yeah, just follow the guidelines and be safe. I choose to pick this slide. Normally, this is something that we should present, and I hardly do it, but I think it is now really something that that drove us in the last 180 days when the pandemic started, because we need to be there to enable the scientists to do their work. That means we have a scientific heart, but we are not doing the science ourselves. We are developing the techniques and the instruments to actually do the real work somewhere else. And in these pandemic situations, I think it wasn't more true than ever before that this is something that we should do. We have to fulfill our scientific needs, and we have to help the people out there to actually do the work and make sure that we get a better understanding of who is infected, what can we do, what is the virus about, what do we need for vaccines, what do we need for therapies. So it was a challenging time, and I will highlight a few things that we addressed during this pandemic, and I think it comes down to two important things. One is we had to deliver, we had to stay open, because everybody else was expecting the material, the instruments, etc. So we couldn't say, hey, sorry, we are closed, we can't do anything. And secondly, and that's also what the previous speaker said, there's no time, and you need to have the scale. You need to produce, you need to be there. So I will highlight a little bit about that, but keep it short. So one thing first, we understood from day one, and it was really helpful that the strategy was very clear from day one, is we need to make sure that our own staff is safe, but that we can keep our operations continuation, that we can keep producing, that we can support our customers, because if we shut down, the process stops. So we had a process in place already for quite some years. When we look at certain processes and projects, what can we improve? It's called PPI, Project Process Improvement. And that's the model that we also used when the pandemic started, because we needed to have one centralized approach. I've got 75,000 colleagues, but we need to have one clear communication strategy, one strategy. How are we going to overcome this? But also from our colleagues' perspective and myself, we didn't know. For us, there was a lot of new information. So we invested a lot of time in training the people, educating the people, but also in communication. What's going on? How do you stay safe? How can we keep our operations open? But how do we work from home? How do you manage that? I mean, we are now all specialists in Zoom and Teams and WebEx, but 7 months ago we weren't. I've never seen so much children from my colleagues before, or walking a dog suddenly in front of the camera. So it's a really different, and we had to get ready to that. It helped. And one of the things that we achieved, and I think the colleagues over there that you see on the picture should be really proud, that even in the worst times of the pandemics in Italy, we could stay open. We could stay and support the customers that were in need of the tools that we were providing. Just to give you some thoughts and feel about the scale and the capabilities that we had to look at is on 3 areas. One was around global testing and sample collection. I think in many states around the world it was, we need to test. But in order to test, you need to have the equipment, you need to have the materials to do that. So suddenly you have to produce massive amounts of tests. I think that right now there are hundreds of projects related to COVID therapies, vaccines, but to do the research, you need certain material, you need certain equipment, you need certain tools, you need certain expertise. How do you get access to that? How can we help? And how can we keep people safe? not only from our own colleagues, our own staff, our own production sites, but also in the field. There was a shortage on material. How do you overcome that? How do you work with that? And I want to share a little bit on what we did and how we overcome that, especially making sure that things were available and how to develop these things. Let me say, to make it a bit alive, from early January, our colleagues in China responded to the virus. We had to mobilize 75,000 colleagues to see what can we do, what can we actually deliver. And as already mentioned, genetic sciences was important. So that department worked on getting the test kit available. But that needed to be an assay that was sensitive and specific. So first we needed to do that. We then look at the pre-analytical part we had internally to discuss with the sample prep team within our biosciences division. Secondly, you need to see, okay, but how are the internal controls? How are the standards, etc.? And we had to go to our clinical diagnostics division. And everything needed to be ready to have early approval regulatory in March. So in a period of 8 to 10 weeks, we had to develop this, but also make it ready and making sure that we complied to everything. Now, once you have the test, you still needed to discuss for regulatory and the commercial teams, how are we going to spread this around 50 countries? That's a challenge on its own. And once you have that, and you have the approval, and you still need to get it there. So you have the supply chains to actually make sure that we scale our manufacturing, but also the supply chain is ready to actually deliver. I think the word sleepless nights is here also very true. It was a challenge. Of course, we did our best and we managed. These are the things that we had to deal with, and it was quite a specific situation. Even going into more detail, this is a timeline overview on what happened from the diagnostic test point of view, and then we only talk about qPCR because I know there are other alternatives. So if you see on the right top, you see that our emergency use authorization was granted by the FDA on the 13th of March. It was actually approval for one workflow on one qPCR, one type of system. But that's not enough to have the scale that we needed because there are certain types of qPCRs around the globe available. We have a whole series of instruments. So we needed to make sure in the months afterwards that we could scale and say, hey, we need to have the approvals for the other instruments as well. So there was a lot of work going on in getting those approvals, having the relationship with the different parties to see, okay, this is what we have, this is what we would like to do, and we need to scale. Now on the bottom, you see also that required capacity, and the test was developed in March, and then we started producing. So completely new test, 5 million tests a week. And at that time seemed a lot. But now we are already aiming for 20 million tests per week by October, just because the demand is so high. Everybody needs to know, do I have it, yes or no? Every country is working on it. So we needed to scale. That was on the kit side. If you look at the instrument side, then you can see actually what we normally produce from a qPCR instrument point of view in a year, we now had to produce in a quarter. So also there, we really, really had to pull our strength together and say, hey, how are we going to help the community? How are we going to do this? And it was really about scale, capacity, and making sure that we could deliver. So yeah, quite a journey. Last slide, example. What else where we had to deal with is that not only on the instrument and the kit side, there was an increasing demand on the needs of testing, but you also need to go to the patient and actually take the sample. But then it requires viral transport media. You need to put the swab into something to make sure that it's isolated, controlled, and everything in the right way. But also there, of course, there was a spike in demand. What do you need? How much do you need? In April, we decided, okay, the demand is going to be huge. We need to produce. It helped that we won a contract by the US government. But then it was decided that we invest a lot in the Lenexa side in the US, where we expanded the facility, hired 300 new people to scale a production that we now can fill 8 million tubes a week, just to make sure that we can follow up with all the testing demand that there is around the globe. That also gave us a blueprint on how to do that in Europe. So currently we are looking and doing the same in Perth, Scotland. time is really precious. So a week or 2 weeks is a lot of time in the pandemic. So we are pushing hard to make sure that actually we can still keep enabling everybody around the globe to do the test and to do the diagnosis. I hope that gave you a bit of a glance on how we experience it, and yeah, look forward to discuss, but also really look forward to have virtual real meetings in the near future again and shake hands. Thanks very much. I hand over to you, Valentin.
Thank you, Erik, for the wonderful presentation. It really concisely demonstrates your sleepless nights. And I think all of us here in the room share this to a certain extent. Producers, dealers, and doctors and scientists in the lab. So I don't believe this will end soon, or at least nobody can say, because since the beginning we've heard all kinds of expectations and prognoses, lots of them based on common sense and past experience, but it seems that this virus is quite tough. So I think it's here to stay for at least the next several months. Now I give the floor to Dr. Zamfirov, who will share his view and an outlook on NGS as a technology in the context of all the other available technologies on the market. So the floor is given. Dr. Zamfirov, you can go on.
Thank you. Thank you, Valentin. You see my presentation?
No, not yet. Still not seeing it.
Should I start the video maybe, or?
Maybe in the share, with the share button.
Yes, sorry. I forgot, yeah. Okay, I'll stop this video, share button, and then go here. Okay.
Yes.
Thank you very much for reminding me. That is my first time experience also for such a type of presentation. Okay, thank you very much. Short introduction. My name is Theodor Zamfirov. I am the CEO of the company Elta IT. We are official Illumina channel partner for 9 countries, and we have our own sales and service offices in most of the Balkan countries, plus Georgia. Now, you know that the virus called SARS-CoV-2 is the responsible for the COVID-19 pandemic, and that's a novel virus pathogen which is identified in China in the late of December. And what kind of test we have to identify this virus is, of course, the golden standard RT-PCR we provide. I will talk more about the NGS, which is a new technique. Next generation sequencing has the unique ability to support the scientists, researchers, doctors, and public health officials, and healthcare professionals also in the front lines. It helps understand the transmission, the roots in the transmission, the comorbidities. So if you have present not only the virus, but also maybe some other pathogens in the same sample, then you can see the mutation rates over the time. Then you can serve as a foundation for vaccine development, some different therapies to see if some resistance appears due to mutation to applying therapies, and ultimately helps predict and prevent future outbreaks. So this is the molecular part of our test. Then we have the screening, so-called screening test, which I divided to 2. We have the antigen rapid test, which is a chromatogenic test. It's in different format, in cassette, in strip. However, it's used for rapid screening in schools, in in a big stuff which works somewhere, and this is for rapid identification, and also antigen for combo, because we have unique system. Now it's running in the biggest hospital in Bulgaria, which can detect 11 pathogen from one sample, and that is very good clinical approach to see if it's not COVID, what exactly is in. So we have already some very interesting examples, like they waiting COVID, but it's streptococcus, it's not COVID. So that it's definitely critical to define the right therapy to the patient. Then we have another serology test, which is rapid, again, serology with without goat, cassette type, for quantitation of the EGM and EGG, and serology ELISA test, which is quantification of the titers of the antibodies present in the sample. And that diagnosis is helping in some phases of the disease where the upper respiratory tract is, you cannot find the virus anymore. So then the blood serology test can help to put the specific diagnosis. And to be complete, there is also method for cell culturing of the virus and microscopy, but this is without any clinical significance for the moment. I'm going to the molecular methods, and depends of what you want to achieve as information. If you want to have a simple detection, yes or no, you can use the PCR, of course, the qPCR, but then you have a lot of other information which is important, and it is good to have back to the PCR with 3 approaches. This is the metagenomics approach, the amplicon approach, and the target enrichment, which we have a respiratory virus oligo panel, which I'm talking to show you for very short time, because my time is very limited and this is hard to explain NGS. This is such a technique so quickly. However, I want to put now the target enrichment work from Illumina, which detects coronavirus strains with equivalent accuracy to the shotgun sequencing. This workflow combines Illumina Nextera Flex for enrichment, which generates targets NGS libraries and a newly available respiratory virus oligo panel. This panel can detect up to 40 pathogens, and in the next slide you will see some of the viruses which can detect, and it's present on the sample. This kit always was successfully used in our National Center for Infection and Parasitic Disease in Sofia, and soon they will go with interesting results to publish. I will not go to this, but just to say you that we have this approach, and this is an approach which is compatible with our benchtop sequencers as iSeq, MiniSeq, and MySeq. And this can be analyzed, and data can be directly uploaded to some application. If you heard about GIS8, this is open database and NextTrain, which shows you real time how, for the moment up to the day, more than 107,000 whole genome SARS-CoV-2 sequencing had been uploaded. So you can track the spreading, and you can see the evolution of the virus, with different mutation, and clades appears already, and different strains already, and genotypes already discovered. So in order to address and different initiatives, medical initiatives and epidemiology stuff. So here is the viruses in the right. You can see alpha and beta coronaviruses plus SARS-CoV-2. You can see also, DNA viruses like adenovirus, and more and more and more here listed. You can visit the website and see in details the pathogens which can be detected in one sample. It's going to be used with like this. So you go to extract the RNA, you go to proceed with copy DNA, then extract only with the probes, which is dedicated to these pathogens. If they are present, they will be enriched, and then you can sequence. The third approach, as I mentioned, is the amplicon. This is quite new test. This is made for diagnosis of the COVID-19 present in the sample, and it's have authorization, emergency authorization from FDA in the US, and also is used in Europe. For the moment, it's limited because you need to have the biggest sequencer machine, NovaSeq 6000, but you can achieve up to 3,072 samples per one run, and to complete all this run in 12 hours. And then you have such a big amount of data for so many samples. You see that the problem and the need of laboratories to expand their capabilities has some limitation. You cannot put 100 of real-time PCRs in the one place, so you need one machine to make to be more with bigger throughput, like this machine I show you here. Also the stuff, you cannot extend too much the stuff, etc. So 98 viral amplicons, 11 human mRNA controls inside, 3,072 samples, as I said. It detects 98 targets, I repeat, it's quite comprehensive, and detect all the genetic material on the virus. How it's done, you again extract the RNA, make the copy DNA conversation, then split in two pools, and you make the amplicons here by PCR, then pool again, and add the internal and external control, make the pooling with the indices, this is the barcoding, each sample, and then you put in the flow cell and go to the sequence. This takes 12 hours, as I said, and this is the indices, that means the sample, so up to 4 to 96 samples, 384 samples per one pool, and one pool is for one lane. This is the flow cell, consists of 4 lanes, as it is visible here, and then you go and sequence it. So 2 flow cells per 1,536 samples achieve 3,072 samples. You can run either 1 or 2 flow cells, depends on your needs. The analysis is going to be done through Dragon server and software, which you can have it on place in the lab, or you can use it base cloud in Illumina base space. You achieve information for viral detection and mutational identification. What this is the results, how is valid the results. You have to have controls, first of all, for the sequencing process, so cluster passing filters, low error rate, positive and negative controls. Then you have to have more 90% of the viral sequencing. If it's available, you go to make epidemiology study and other study of the sample. And also you have to have in mind that this test detects entomology regions, and it's not fully excluded from the detection. So that is the all parts which have to be ordered. So everything is here from the beginning up to the end. So it's end-to-end solution. And of course, you can go and see more information about this product in our partners portal and website. So thank you very much, and I will be glad to take your questions.
Thank you, Dr. Zamfirov. I have a few questions, but like I mentioned, we will address those at the end during the Q&A session. And now is the time to give the floor to Professor Argirova to share her own experience about this epidemic. And she will talk about tackling the problem and the natural limitation of the diagnostics of COVID-19, which is that sometimes there are false negative results from the PCR testing. Professor Argirova, the floor is yours. Please go ahead.
Thank you, Valentin. Thank you, everybody from this biologist organization. I feel there is a lot to speak about the diagnostics of SARS-CoV-2 infection, but I have selected some questions which are very often met or unmet, both are regular, in the clinical practice, being a clinical virologist in the private hospital Acibadem CityClinic Tokuda in Sofia. So, you know, there are a lot of problems for the clinical virologists during this pandemic. About false positive results, they lead to unnecessary quarantine, and I'm not going to speak about that, but I would like to stress on the false negative results, and especially false negative RT-PCR results, because these results are dangerous, especially in asymptomatic persons, because this does not lead to isolation, and infection of other people is possible. Also, this question is very important because we very often have some disputes or discussions with the clinicians, radiologists, and I would like to stress on this. Here I have taken, this is a well-known considerations for diagnosis, and what I would like to stress here once more is first this period between the -2 and -1 week. It means because the symptoms onset, I mean, this is the time you see in this diagram when the red line, the virus isolation is too high from the respiratory tract, but during this time, very often the patient has no symptoms, has no complaints and this is the most dangerous period, as we all of us know. And after that, the PCR is getting positive in nasopharyngeal swab. You see the green and the blue line in nasopharyngeal swab. Then somewhere after the end of the week 2 or the beginning of week 3, we see that this PCR is very often going down, especially in nasopharyngeal swab. So all this is important because this means just when should be the right time to take the sample and to detect the RT-PCR in it. Here I have stressed on some factors potentially leading to false negative results in symptomatic patients. It's important because the asymptomatic patients, this is another chapter. And first, of course, everybody of you know about the poor sample quality due to improper sampling or handling, especially due to transportation. To avoid this, in our hospital, we have only specially educated persons who perform collection of samples. Then the second is the inadequate timing of sample collection, and that's the reason I have shown the previous diagram. Too early or too late, when the PCR is positive or negative, you should know this timing. And again, I'm saying that about the end of the second week after the symptom onset, it's for sure that the PCR should be positive. I I'm going further. Low viral load, especially soon after exposure to the virus or after the second week of symptom onset. Of course, inhibition of amplification or factors inhibiting amplification. Here, the role of internal controls is extremely important. Also, another cause for the false negative PCRs should be viral mutants if available. Especially, I have stressed here on mutants in S gene, but not only, also in N gene and in the other genes, of course. A lot of mutants have been already described. But what I would like to stress here is that recently more PCR tests targeting 3 genes appear at the market, and their interpretation sometimes is difficult. Is it only the mutants to blame here? And this is especially when one of the genes of these 3 genes is not expressed. It's difficult sometimes, this interpretation. Another factor is which is important also to come to such a conclusion, that one or more negative results do not rule out the possibility of SARS-CoV-2 infection, especially if the patient is highly suspicious for these infections, having in mind symptoms, radiography. So here I would like again to remind that the clinic is extremely important, and we should see the clinic first, and after that to try to confirm with the other methods. Again, very often there are false another factors which another things which we can do in such patients who have the clinic symptoms or radiologic symptoms, but negative PCR test. What we do and what to do in such patients? First, try to collect additional specimens from the lower respiratory tract. For example, we got PCR positive on the 3rd day or once on the 3rd time of testing in the hospital, and only from a bowel sample. So we couldn't, about this patient, we couldn't obtain a positive results from the nasopharyngeal swab. Also sputum in cases with productive cough are useful because it is considered it comes from deeper parts of the respiratory system. Then again, an advice to follow the individuals living with the patients. We have 2 cases with full clinical and image evidence for infection, but PCR negative, and 2 or 3 days after the admission to the hospital, their girlfriends showed PCR positive result. And we consider this as a good epidemiological evidence for infection. So another reason for the false negative RT-PCR should be the so-called prolonged nucleic acid conversion in these patients. Here I have stopped my attention on an article published, I think in March this year, where the Chinese colleagues are discussing about so-called turning positive by RT-PCR for the infection after 2 consecutive negative results. And this may be related to false negative RT-PCR tests and prolonged nucleic acid conversion. Also, this nucleic acid prolonged nucleic acid conversion would be with low viral loads. The patients studied in this article were all of them with mild symptoms. And I would like to repeat again, what does it mean, nucleic acid conversion time? This is the time defined, this is the period from the date of symptoms onset to the date of first negative PCR test result. And this time is different in different patients, but we can say that in most patients, this time is about 18-20 days. Some authors even speak of less time. But in this article, the conclusions which were made is that younger male patients, 43 to 65, had experienced shorter nucleic acid conversion time, 21 days, vice 36 days in older patients. So it means that some patients demonstrate a prolonged nucleic acid conversion regardless of the relief in symptoms or radiology. Why is this important to be known? This is because we know, all of us know, that the current guidelines suggest 2 consecutive negative RT-PCR test results as one of the criteria for discharge. Also, all of us know very well that somewhere in the end of March, beginning of April, some scientists, virologists also, have proposed the quarantine time after discharging from the hospital to be prolonged just because of such results, because such results have been described also in another works. So such patients are often considered with recurrence. This is not so. This is the same infection, but there is such prolonged nucleic acid conversion, or even as a second infection, but we know very well that the second infection should be proved by in other methods. Now, I would like also to say some words about this. What characteristics are missing in RT-PCR testing of SARS-infected persons, SARS-2, of course? We believe it is important to measure the so-called pre-test probability. It means the individual's chance to be infected in asymptomatic persons. This is important because now we are under pandemic conditions and because we want to contain the infection. So that's why it's extremely important to know first the share of these asymptomatic persons. So last article say it's not more than 20%. Earlier the percents were bigger. At large tests, this pre-test probability depends on sensitivity of PCR tests, but at the same time, we know very well that not all PCR tests are of high sensitivity. Some of them are with lower sensitivity or even differing in sensitivity from one to another lot. But the pretest probability does not depend only on the sensitivity of the test, also on some symptoms if the person has contacts or exposure to the virus, local SARS-CoV-2 prevalence, etc. So test sensitivity in asymptomatic patients seems very important, and this sensitivity would be important not only for analytical, but also for clinical and epidemiological purposes also. So today, it still remains a difficult task to define the pretest probability in asymptomatic patients, and this seems very important for the further development of the pandemic. So thank you for your attention.
Thank you, Professor Argirova, for the wonderful presentation. Now I suggest we go and discuss some more details and some of the questions to the panelists' talks. My first question is to Dr. Lezhava from the Genomic Institute of Singapore. And my question is about, you mentioned that in Singapore you have developed assay for isothermal amplification. And I would like to ask you, in your experience, is isothermal amplification very sensitive to the specifics of the pre-analytical process? The sample collection, the transport of the samples, the extraction, what is your observation in this regard?
Asking that question, so I don't. Can you hear me? I'm sorry.
Yes.
Okay. So major thing that is based. So major issue with isothermal amplification is that it is quite well known that in most of cases it is not as sensitive as PCR. So PCR is more sensitive. However, isothermal amplification has own advantages like it's extremely fast compared to PCR, and also I think it's. I would say that. And it's going on one temperature, so we don't need any sophisticated instrumentation like PCR instrument, or just can be done on regular kit and so on. On the same time, yes, how sensitive it is. Like any other test, PCR test, of course, isothermal amplification based also on activity of polymerase, which is a little bit different. Instead of exo activity, it has stun displacement activity. However, as other polymerases, it is also sensitive to inhibitors. So I think, of course, in most, I can also comment on the PCR kits itself. Some of them are more sensitive, some of them are more resistant to inhibitors and can overcome sometimes this inhibitory issue, which is very, very common for viral transfer media and also sample itself. So again, yes, definitely it is. However, and in this case, many efforts are coming to come up with, because it is a rapid test, and to combine this rapid extraction of direct PCR, it's very, very advantageous. But at the same time, yes, definitely, if there is no proper action of against inhibitors and so on, I think we are also giving up for another sensitivity related method. So, and this is something that can also cause the false negative results, which was very well described by colleague in the previous talk. So yes, definitely we need to take care of taking consideration those effects where before we are running one or another direct PCR or direct isothermal amplification and so on. So yeah, thank you.
Thank you, Sandro, for your answer. I would like now to ask Erik something, which is what is your experience in Thermo with automation in this in the COVID situation for those last, let's say, 6 months? Do you think the scientists' view on automation has changed, and how does a company like Thermo keep up with what everybody sees as increasing demand for platforms, for consumables, even plastics? What is your view on that?
Yeah, thanks very much for the question, and just a comment to the previous speaker. I will also share the comments on qPCR test, etc., to my colleagues, just so she's aware. I think automation, when you need the scalability that is currently asked in some places, you cannot do without automation. The only issue was we needed to develop something specifically for this case. So we did that. I think it took us 140 days, but the system, I think it's launched now 2 weeks ago. We have now a fully robotic automated system to do 6,000 assays in 24 hours. We see that there is a need. We see an increasing demand, but it also requires specific manpower. It requires investment. It's not something that you can easily do out of the box. So it's always a balance. What do you need? What does make sense? You need to look at the specific situation, but we see that there is an increased demand. And what we are doing, and it's not only on the diagnostic side, but also on the vaccine side, the therapeutic side, we constantly are trying to monitor and discuss with our customers, what do you expect? Where do you need help? What kind of capacity issues do you foresee, so that we can schedule our production? Because we cannot be too late. So we need to constantly make scenarios. What do we expect? What is the market expecting? What are our customers expecting? But it's sometimes, information can change within a week. So we need to be on top of it and listen good to the advice that we get, and to make a plan as good as possible.
Thank you, Erik. Thank you very much. I would like now to ask something to Dr. Zamfirov regarding the application of NGS. Personally, I see one of the main advantages of NGS technology in providing additional information about the sample, which is both information about other infections, but also information about the genetic makeup of the patients. So my question, Dr. Zamfirov, is before such a large-scale metagenomic approach to testing, what are the main challenges and limitations, and how do you think they translate to smaller markets and economies like Bulgaria?
Well, good question for small markets. So, okay, in the science, as in the sport, there is no small and big nations, right? If you are specialized in some direction, you can create good scholarship with professors which can go deeply into some kind of the fields. So shotgun metagenomics, first of all, initially was, in principle, it's a high throughput sequencing method, which is, first of all, invented for identification and genotyping of microbial communities in a given sample. Prior knowledge of what microbes has exactly in there. So the comprehensive examination of all organisms in a sample is a crucial when you want to discover a new pathogens like SARS-CoV-2. For example, with this approach, you can do it. Secondly, the shotgun technique, when you have, in principle, you sequence what is inside in the sample, and then your sample was compared with the readings with some database. In the case of Illumina, Illumina is using third-party database, a company called ID by DNA, which has data sets of 35,000 viruses and more than 13,000 bacteria. So you can determine in your sample what is exactly the pathogen. Secondly, there is a co-infection with COVID with something else? Third answer of the question is, if it's not COVID, what is inside? And this is crucial because nowadays society has a lot of methods to educate themselves. They need answers. They don't need a test, they need answers. If I don't have COVID, what exactly I have? So laboratories is a challenges. This is a challenges and pressure from the society to give answers. So this approach is giving answers. And secondly, if we want to dive even more deeply, you obtain from the nasal swab, you obtain also RNA from the epithelial cells. Then you can see the expression of the genes of the body, of the human expression of the genes, how the body reacts to the inflammation. Then you can have the immune system, the single cell, etc. So the single cell examination, sequencing also provides information about what part of the immunity is activated, etc., which is very valuable, of course, information for different type of research. And in fact, that shows show you how to go with surveillance in the epidemiology, because nowadays with the mutation type, we can say that there is no mutation which can increase the pathogenicity. Okay, there is suspected that they increase the infection rate, so they can infect the spreading of the viruses more with one mutation, particular mutation in the spike protein. But in the pathogenicity, so this information gives us the importance of the other measurement, the masks, the washing hands. So it's more and more valuable, the viral load, how many viruses you take into this infection. That will be crucial for the pathogenity of the virus, because the virus is one and same, showing from the data here.
Okay. Good. Thank you very much. I think coming back to also to Erik and his experience in biobanking, and to something that I have discussed with a lot of specialists, there is also a great need to collect a lot of samples at different stages of the infection, and I want to follow up on this concept with Professor Argirova, and actually to ask Professor Argirova, what do you think? Because you said one of the most problematic things right now is to collect data about the asymptomatic patients. What do you think is efficient strategy to get to asymptomatic patients and to collect more data about them and about their status?
Okay, thank you for your question. It's a good question, but it's very difficult to answer this question, because the only way is to notify the contacts. But it sometimes is not possible because of we miss these contact persons, or sometimes they simply don't want to give another sample to be studied. But nevertheless, I think that where possible, we should follow these contacts and to see what would be, somehow to monitor what would be the RT-PCR results, and of course, the best of all, qPCR results during the time of this contact or asymptomatic situation.
Of viral culturing. What do you think is the place of viral culturing in, so to say, the national strategy for investigating and controlling such epidemiologic situation?
You were the first part of the question on mute, Valentin, so I don't think we know to who you asked, and we missed the first part.
This is to Professor Argirova about viral culturing. What do you think, Professor Argirova, is the place of viral culturing in a national strategy to investigate and to fight against this epidemiological situation? Because not many people lately mention viral culturing, but what is your opinion about this?
Okay. Viral culturing, or as you say, this is very important for us virologists because of several reasons. First, it's very important to measure the infectivity of the virus in an appropriate viral culture. So this is the first. We could find a higher or lower infectivity of some mutants, for example. So it's not only to see what happens in society or in different members of the society, in different patients, but also to see how the mutants change their infectivity. Also, another characteristic, transmissibility. For example, what comes me in mind just now is that today this new mutant, which is actually this G mutant, so-called G mutant, D164G mutant, as if it has no higher infectivity but higher transmissibility. So all this could be very well established in tissue culture. The second reason to have viral culturing or tissue culture with the appropriate virus strain is about the experiments on inhibition of viral replication. This is a good model system to see what would be the possible inhibition by some substances differing by more or less chemical groups, etc. And this should be very interesting and useful, I think, of good benefit. This is the second, which would be important. Then, of course, the viral culturing is very important when we speak about viral inactivation or even disinfection. This could be very well proved in such appropriate viral culture.
Thank you very much for your answer. I have one question, Erik, as quite experienced person in biobanking, what is your opinion about the importance of sample collection at regular intervals? And maybe here you could also mention your view about the place of serology in the diagnostic landscape. What do you think on this?
Yeah, thanks. So I think, first of all, from a biobanking perspective, it is the research community needs positive samples to test certain vaccine candidates, all kinds of things. But next to the positive sample, you need also the background of the patient. You need to have the treatment protocols, the disease severity, the clinical symptoms, all that kind of information you actually need to collect. And of course, you need to make sure that you have consent from the patient to use the material. In some cases, it's also very helpful if you have the same material and data from the patient when they were still healthy. We all know that in the northern part of Italy, it was the pandemic was quite severe. But between summer and Christmas 2019, they did a population study in the area of Bergamo, and there they could go back to certain data and samples from the time that the patient was still healthy and see, hey, do we see a correlation that some diseases they already had or some other issues? And that was really helpful. So I think it's really important to collect the samples in the correct way, maybe also for the future. And there, serology can play a role. If we look at the false negative presentation from the professor, that you have a certain time window, and with serology you can have a longer time window to look at certain samples. I think the infrastructure to collect the sample is something that we are more used to than taking the swabs in parking spots with teams that are not the experienced as they should. So there are all kinds of new ways I think we can detect the virus in a later stage and also see how it spreads across the population, and serology could be a good option going forward in parallel with other solutions, but it could be one of them.
Thank you, Erik. I have a few more questions, but because we are running out of time and it is already shortly the next panel should start, I will address the remaining questions to all of you additionally, and then with the recording of the panel, we can provide also your answers and additional viewpoints to all the attendees and interested persons. And with this, I would like to thank all of the panelists for this session, and to express once again my gratitude to the organizers. I'm looking forward to the next digital event